So I was talking about the gene expression control. Yes, the turning on your gene expression, maybe starting the transcription and then continuing with translation, those processes are highly regulated in your cells. Okay, so it could be regulated at the beginning of the transcription, or it could be regulated at the maybe processing of RNA. It could be regulated at the translocation of the RNA, mRNA into the cytoplasm, or it could be regulated during the maybe translation, maybe initiation of translation, or during the translation, or modification. So the gene regulation happens at multiple steps, okay, in the transcription, translation processes. So here's an example how alternative RNA structure adaptation will regulate the gene expression process. Okay, so this mRNA, so it's coding probably important protein. Okay, so you don't have to know what that protein is. It's making a protein that helps the pathogenicity of this virus. So I guess it's bacteria. I'm sorry, not virus. So Listeria monocytogenes. Okay, it's an enzyme that's made by this. Anyway, so if this RNA adapts this conformation, so let's say there's a pre-mRNA, okay, and there is that, so it's probably ahead of the gene coding sequence. It's a ribosome binding site. So if this mRNA is available, so the ribosomes will bind and then maybe start the translation. But if this mRNA adapts this conformation because of the extra sequence, and then there's that loop, a hairpin loop is formed, okay, then this ribosome binding sequence is not available for ribosomes. Large subunit, small subunit cannot hold onto the sequence. So if that happens, then translation is not going to happen. So no matter how many of this mRNA is available, this mRNA is not going to yield the protein products, so no translation happen. Okay. However, under certain condition, the condition is what? High temperature. So let's say this infectious bacterial cell is infecting a human, for example. So the infection normally causes what? High fever. So if there's high fever happening on the body, the high fever temperature will break up this loop, especially that around that area, because the hydrogen bonds are broken up. So then this secondary structure of the RNA is changed to this form. Okay, and then if this conformation is made, then this RNA binding and ribosome binding sequence is now available for large subunit and small subunit to hold on to. So ribosome binds. If the ribosome binds, then they run through the rest of the mRNA. So the translation will continue. So if the translation starts, then what happens? The proteins are made. So the enzymes are made. So that will contribute more infection of this the pathogen into the body, right? So what the importance of this diagram is what? The adoption of RNA structure into this form versus this form, it kind of controls the translation process through the availability of the ribosome binding sequence. Okay, so it's a kind of unique, novel mechanism where RNA structure change, mRNA structure change controls the translation of a protein. Makes sense? And then RNA can fold up into a complex tertiary structure. So here's another example. So the tertiary structure of RNA here and here. So they can adapt different tertiary structure based on the hydrogen ions availability to protons. Okay, so proton changes, so that's the pH, right? pH depends on the pH of the environment. This RNA tertiary structure or this RNA tertiary structure could be made. Okay, then how those different RNA structure affects maybe the gene regulation. Think about that virus, the murine leukemia virus. It's a unique virus. They carry RNA genome. So that RNA genome can adapt different structure. Okay, and among those genomes, they have important functional protein coding genes. Call them those GAG protein coding gene. Sometimes that GAG protein is expressed alone, or it could be expressed as a kind of fusion between GAG and polymerase together. So anyway, so the switch of this RNA tertiary structure, okay, is controlling the gene expression, either just GAG only, GAG protein only, or it could be made as a GAG and polymerase together. Okay, so 90 to 95% of the case they adapt this structure. The RNA structure adapts this. If that's the case, then the translation stops right there because there's a stop codon. So the only GAG protein will be made, translated. Okay, but under certain condition. Okay, what is the condition? It's change of the pH. Okay, so responding to the proton level. Okay, so maybe if the proton level is changed significantly, then this tertiary structure is adapting that, which is different structure. If that happens, then what? So these ribosomes can run through this stop codon. It doesn't stop at the stop codon. Instead, it can go through that, and it continues the translation. So not only GAG protein is made, the GAG is fused with other codons after this. So it's actually GAG and polymerase fused together. The complete protein, larger protein will be made. So polymerase is of course needed to further replicate RNA genome and make more of these viruses instead of a host cell. So the switch is important for the further, infection of the viruses. Okay. Then finally, some RNA molecules exist as enzyme. Okay, they serve as enzyme like role inside of the cells. We call those RNA molecules as ribozymes. Okay, ribozymes are enzymes made out of RNA, ribonucleic acid. Those are ribozymes. Okay? So a lot of RNA serve as ribozyme function. They behave like biological catalyst. Okay? Example is here. So RNA is a P. It's an enzyme. Okay, it cleaves the leader segment of the 5' and the precursor tRNA to generate the mature functional tRNA. So remember tRNA, which is carrying out the important function during the translation. Okay, when this tRNA is made through a transcription, it's actually made as a larger precursor, and the pre-tRNA is modified. So its 5' end is cut up. That cut up is made by this unique enzyme called RNase P, which is made up of another RNA molecule. So that cutting, okay, is made by the RNA portion of the RNase enzyme. That's what it means. So RNA can cut other RNA. So that's the example of a ribozyme. Okay, RNase P. So it turns out RNA, there are a ton of RNA molecules, different types of RNA molecules exist inside living cells. Many of them function as like ribosomes, maybe tRNA. On top of that, some of them may function as enzymes. We call them ribozymes. Beyond that, there are a lot more different RNA molecules that can serve as different functions. We will learn a lot more about RNA molecules through this class later on. Okay?